Okay, and we're now going to go to our last but not least presentation from Yves Sabbagh from INZ-701. And so, Yves, the floor is yours. I see your screen. Okay, great. Yes, yes.
I'm going to try this one more time, but I might not be able to put it on full screen. Or
okay, that'll be okay. Or you can email it to me and we can present it.
Okay.
Ah, how's that?
If not, I'll email it. Yeah, that's fine. Okay. Thank you, everyone. Sharon, thanks again for the opportunity. And this has been a great meeting. I've also learned a lot listening to others. I love the collaborative nature of the meeting. As a disclosure, I'm the Chief Scientific Officer at INZ-701, and I'll present our phase 1/2 trial on INZ-701 that we've initiated in 2022. Next slide. So again, Hubert showed part of a more expanded view of the pathway. I won't belabor the point too much, but the enzyme that INZ-701 is really focused on is ENPP1, which is the major enzyme that generates PPI from ATP. As the others mentioned, there's a lot of ATP inside the cell. We need the ATP to be on the outside of the cell to be able to convert it to PPI by ENPP1. There are also other sources of potential PPI, but the biggest amount of PPI comes from what's generated from ENPP1. And we are also focused on calcification disorders, and there's another disorder called ENPP1 deficiency or GACI type 1 and ARHR2. These are patients who have really mutations in ENPP1, and they have really about 80 to 90% less plasma pyrophosphate levels than the healthy population. In ABCC6, as it's been reported, and we've seen it as well, is between 40 to 50% levels. And that's because they do have a functioning ENPP1, but they don't have the ABCC6 is mutated to bring out the ATP, but there are other sources of ATP that could be also available. And ENPP1 is stuck to the membrane usually. Our molecule, which we call INZ-701, is basically where we take the outside part of ENPP1, which has where the activity of the enzyme is, and this will be able to circulate throughout the body and find sources of ATP that could be available, and that's how we're going to be able to bring the levels of PPI up. We've done a big collaboration with Lily as well as Yoni. Yoni's been involved in every aspect, I think, of research and drug development in trying anything for the patients, which I think is great, and we're all going to miss him. We published a paper on the data on the mouse last year that shows that with INZ-701 we can prevent calcification by giving it in the vibration and increase PPI. So our enzyme that we're giving is to replace an ENPP1 deficiency, basically ENPP1. In ABCC6 is to have it circulating and find other sources of ATP that are not coming from ABCC6, and to be able to convert ATP to PPI. So we have initiated a phase 1/2 trial. So it has two components. It's again the primary, a bit low what Hubert just mentioned. You have to establish safety first. So this is a safety as well as understand your properties of the drug in circulation. How long does it last? And other parameters. And of course, we're also measuring PPI. So being that our trials are a phase 1/2, what it means is we have a phase 1, which is for 32 days, where we measure the INZ-701 is given for one dose first, and the patients are followed for a week. And this is to establish how long the drug can stay in circulation, because you get PK parameter, how is it accumulating? And then we start chronic dosing from day 8 to day 32, twice a week sub-Q, so under the skin. So this is in adult patients with genetically confirmed mutations in ABCC6 and have PXE. And after the 32 days, we go what's called a phase 2 trial as an extension. And there patients keep getting receiving the INZ-701 for twice a week for up to 48 weeks. And after that, they continue, the patients are continuing on the drug until the drug is either approved or the development is stopped. So it depending on the outcomes of the trial. So we are in all 3 cohorts now. We have 3 patients per cohort that are being dosed. And the cohort 1 is the data I'm going to present today, which is the day of 32 days. What I could announce is, which we announced last week, the company is on February 16th. We are having an event to present the data for cohort 1, 2, and 3 for the top, showing the PPI and the safety and PK/PD parameters that we have right now. Obviously, we don't have the full data set. Like I said, this is up to 48 weeks. Cohort 3 is only been started enrolling and is at the end of last year. So that's gonna basically at the end of this year, we'll have the full data for all 3 cohorts, but we will be showing what's called top line, which is some safety parameters that how PPI is increased, but in all 3 doses. Today, I can only show you for cohort 1, which is 0.2 milligrams per kilogram. And in those extension, we have baseline, and just like everyone else, we're trying to learn on the progression and what could be the important endpoints that we can eventually do what's called the pivotal trials, which are the trials that are key to be able to look at these outcome measures where we can have a really clinical, meaningful effect to be able to get the drug approved. And so we are doing assessments in the eye, imaging, and as well as CT for calcification and in the patient at baseline, and so that will be helpful for this. And I think working together is going to be key in learning, and even listening to Christina's talk, I think it's going to be valuable, the work that she's doing as well. So this is the lowest dose that we have, the cohort 1, as we call it, at 0.2 milligrams per kilogram. So as I stated, this is we give the INZ-701 first 1 injection, and we follow it for 8 days, and then on day 8 we start giving it twice a week, which are the yellow dots. So the black dots is just following it after a dose, and yellow is always a sample of PPI before. So one thing to note is that we have this green box. This green box represents 10 healthy volunteers that we recruited to really measure what PPI in our assay is. And what Kern presented this morning, we also have an assay, and it follows a similar principle, the ATP sulfurylase assay principle. But again, to answer people's questions, and as well, we did follow these 10 healthy volunteers, adults, for over 3 days, and we did look at them fasting all day. We did them when they were having regular meals, as well as when they exercise. So we do have human data for exercising. We have we wrote up the paper on what could influence PPI, and I can tell you exercise will bring PPI up significantly, actually. But it does go down after resting, back to baseline levels. What was also interesting to learn is that fasting gives you the least amount of variability, because in foods you have sources of ATP and other sources, even of PPI that could be there, so they can increase, and you get a lot of variability. But overnight fasting with no exercise, we saw that because the 10 healthy volunteers, we have sampled 3 days in a row to really assess how changes it is. And we know fasting gave the least amount of variability. So this is what the patients also their PPI were collected at fasting overnight. And what was really interesting to see is that even after the first injection, okay, here at time 0, within 6 hours, we saw an increase in PPI. And this increase went further to 24 hours. But after that, it went back to baseline. And when we started giving it chronically, we saw a small rise in PPI, but the levels are not high enough. Now, this, like I mentioned, this is the lowest dose. And in the mice, we had to go to higher dose levels to see an effect. And we are going 9-fold higher in our clinical trial. So cohort 3 will it'll be very revealing, presenting the data at the 1.8 milligrams per kg in February. But right now, this is what we could see, that our drug can at least transiently increase the levels of PPI at this dose level. What we also saw is, like, in terms of PK parameters, which is again how the drug behaves in circulation, and we look at the activity to make sure that it is active. What we see is very nice accumulation of INZ-701 with time, which is important, and the half-life of the drug, so it's how long it lasts, was surprising that it was about 5 to 6 days long. And this is suggesting that we could potentially move from dosing this twice a week to dosing once a week. Now we're in the middle of an analysis for the other cohorts to really confirm these effects, and then we do what's called dose simulations that are important to really do the next phase, which is the dose selection of what would be the ideal way, because you don't need to give more drug if it's not needed. So this was exciting for us to see. In terms of safety, for cohort, for the data I'm presenting you today, it's been very well tolerated, exhibited a good safety profile. We haven't seen no serious adverse events reported. We've seen some grade 1 in in this 32-day cohort event that was non-drug related. We do see some anti-drug antibodies that were observed in the subjects, but it was very low titers and it had no impact on PK or ENPP1 activity, which is important to tell us that you can potentially have an immunogenic response, but it's not impacting the function of the enzyme, which is important. And all 3 subjects in cohort 1 did continue into the extension phase, which is now the phase 2 trial. And actually, cohort 1 are going to be finishing soon this year. Obviously, at the beginning of this year, we'll be finishing the 48 weeks, and the other cohorts are continuing. So we are very excited about these. So just as a summary, we did show that we can increase PPI levels in subjects in PXE patients, in this case with functional ENPP1, because we also have a trial in ENPP1 deficiency, which we're also going to be showing data the same day. But PPI did increase in all 3 subjects into the levels observed based on our 10 healthy subjects using our assay. And PK and activity is consistent with what we saw with our other trial in ENPP1 deficiency. And safety up to now has been very good. We've had patients even in the ENPP1 trial that have been over a year now, and they're still on INZ-701, and safety continues to be very good, which is obviously very important for this. So we'll be looking forward to presenting more of this data to everyone on February 16th. The information will be on our website for the links and more on that. And finally, I'd like to acknowledge the research team at Inozyme, as well as all the other components. To run a clinical trials requires a lot of components, clinical operations, to make sure the patient's needs are met. If they have to travel, the CMC is the group that makes the drug. Obviously, the work we've done with Yoni as well as Lily has been very important to do the proof of concept in the preclinical studies. Frank Ruch, Yvonne Nietzsche, also, we've done a lot of work with them on the ENPP1. Sharon, thanks again for the opportunity to be able to have great discussions together offline, as well as at these meetings, talking to other scientists as well as the patients. The patients, obviously, we'd like to thank, who are in our trials. It's a big time commitment, as everyone knows, and we really appreciate. I think that's helping the whole community. We've done work also with Wilco on trying to get some samples to really understand better dynamics of ATP, PPI, as well as other components. And recently, we've also connected with Olivier to also look at other biomarkers and other endpoints And it's been great to meeting more people and expanding our network and helping each other to get to the next phases to help the patients. With that, I will close and take any questions. Thank you.
Okay. Thank you, Yves. The first question in the chat is, what type of exercise increases PPI levels? Does target heart rate need to be achieved and over a certain time period? And does age impact this?
Yeah, so very good question. So we've done it. The 10 healthy volunteers were between 18 and 30. Although I could say we haven't done the exercise above 30 right now. We've done age does not impact PPI in terms of in general. You know, we even had a paper we've done with Frank Ruch looking at PPI in healthy subjects from 0 newborns basically to 18 years of age. It's pretty much of a flat line. It doesn't change that much with age. But in terms of heart rate, the way the settings we did to look at exercise was bringing them up to 80% of their maximal heart rate. It was like they ran on a treadmill, and we did see, and it could practically doubles, close to doubles the PPI. So it is that's the only thing we tested. We did not do longer. We just made sure they reached their 80%, and it was max about half an hour that they were on that.
Great, thanks. And of course, you know, PXE-ers, you saw in our studies and so forth, average age of late 40s, 50s, 60s, and so much older than some of your controls. A question that is probably for you and Hubert, and I'm going to actually take a crack at it and you guys can add. So if the phase 2 is successful in either of these studies, and I assume Nico, who's asking the question, would mean any of the studies, typical phase 3 studies need around 5,000 or so patients to get a drug approved. How does EMA and FDA handle these? So both EMA and FDA have a long history of trying to handle rare diseases well, starting with the Orphan Drug Act 40 years ago next week in the US, and then EMA with some also mechanisms that are quite good. And so orphan drug designation creates a different pathway for these drugs and requires fewer individuals. In some cases, there's a new path where a number of especially pediatric indications that are fast-tracked can go to market with Ns of 1, N meaning the number of people, which is quite unusual but necessary in certain very lethal conditions. PXE, one of the things that we're often trying to tell these regulatory agencies is that there is a burden of disease. And in fact, you will be getting a burden of disease study from us in about a week or two that we're doing across lots of conditions and showing that there is a burden living with PXE. When FDA and EMA look at diseases like PXE and compare them to diseases where a kid dies at 5, they say not such a burden, or cancer where you have, you know, months to live. And what we're trying to say to them is, in fact, there are a great deal of burden with this disease. And so things like that make a difference about how the regulatory agency will consider the disease. And so Yves and Hubert, what do you want to add to that?
No, it's a very good point. You know, I've been working on rare diseases for over 20 years. I was at Genzyme before, so I've exactly what you said. Some did, depending on the rarity of the diseases and how you design it. If it's in adults with placebo control, you can do a 2-to-1 ratio of 2 being on drug and 1 not. And you know, some trials could be 25 patients only, depending on the rarity. You know, it wouldn't be in the thousands for PXE, not from from our perspective at INZ. It's not going to be something that's going to be required, and don't think it's reasonable for as a request from the FDA. And I know they won't do that. So.
Yeah, I totally agree. Just adding to that, certainly it will not be 5,000 participant study in the rare disease setting, and all the regulators recognize the need, the unmet medical need there, and they recognize in the rare disease space, they're not going to have those that number to meet the requirement. But the important part for them is really trying to understand the balance of meeting the need in the setting where there's no available therapy, and what are the data to show that it actually can improve, and particularly improve the quality of life and improving how you feel, how you function, and balance that versus the safety of the particular agent you're talking about. So that's, that is really the balance, benefit and the risk that they need to consider, and we all need to consider.
Other questions?
I see one, the
Okay.
presentation in February. Yes, it's going to be available online, live, February 16th. We'll have more details. The time is still being worked out, but if we go to our website in the investor relations section, elsewhere, and then we'll have a press release announcing more details on that. So, but it is publicly open. It's not a private event, so.
Okay. All right. Thank you very much, Yves. Thank you to all of the researchers here who presented. You can see that the combined amount of work that's going on is quite incredible. A real testimony to the various individuals and institution and company care about PXE and about what needs to happen for you. When I look back to 28 years ago, 26 years ago with our first meetings, when we were barely able to describe, oh, there might be something called angioid streaks and there maybe is some pain in the legs and so on. This is quite the difference over these years. And again, largely because of you, both because of the data that you've been sharing. Kat will put in the chat right now the URL for the surveys. They're absolutely critical for us to amass the data we need, and then also to figure out which trials make sense, and then also your contributions. So we often say that even $5 is really helpful because you can understand that particularly the academic researchers need our help. We have a couple proposals from a few of them here that we're considering right now, and we do want to fund all the research that we possibly can. So so thank you so much for your support.